All right. Hi, everyone. My name is Vivian Zhang, and today we're glad to have Jack Liu, who is the founder of Omicsoft and now VP of Bioinformatics at QIAGEN, to present to you today's webinar. And for today's webinar, we will first turn to the presenter for 40 to 50 minutes presentation, and at the end, we will have a Q&A session. So please post any questions you have during the presentation. And at the end, our presenter will pick up as many questions as we can. If your questions is not addressed during the webinar, we will definitely get back to you with the answers through email in a couple of days. So I encourage you to post any questions you have during this session or during the entire webinar. All right. So let's get started with the presentation. And now I will turn to the presenter, Jack Liu. Welcome.
Thank you, Vivian. Hello, everyone. This is Jack Liu from QIAGEN. I'm very excited to present to you an incoming new release of Array Suite. This is Array Suite 10.0 from the Omicsoft team, which now is part of the larger QIAGEN Bioinformatics group. I hope that you will find the information to be interesting and useful. In addition to the planned features in 10.0, such as cloud-based NAND, single-cell processing, I also will introduce some of the integration projects that we are working on, which I'm personally very excited. So hopefully, we will get into some details, but also hopefully you can get some big pictures of the new release, and I will be happy to answer questions at the end of this webinar. I cannot imagine, but it has already been 10-year anniversary for Omicsoft. I was personally involved in this 10 years, and I was so excited to see every single step of this journey. In 2007, Omicsoft was founded in North Carolina, Research Triangle Park. About 6 months later, we introduced Array Studio version 1.1. This is a single desktop solution, and we won the first contract. We spent a year to release a server in 2008. 2009, we started to provide DEO data service. So basically, we analyze the data for hundreds and thousands of DEO projects, and we analyze this on behalf of our customers. So this is essentially the prototype of the NAND service that we have right now. In 2011, we actually started the TCGA data service because TCGA data was so informative and useful at that point of time. At that time, we just regarded it as a big project, and we analyzed the project. We curate the data. It was the prototype of ENCODE, but we didn't actually realize that there is a need for a large-scale data processing, data management at that time. We did some improvement on this server in 2012, and in 2013, which I personally think is really the breakthrough of Omicsoft. We released ENCODE as a NAND technology. We continue to improve the framework by adding the cloud integration in 2014. 2015, we expanded NAND technology from oncology to non-oncology, which now is called disease NAND. Last year, we released genetics NAND, which means that Omicsoft is now interested in not just the Mercury RNA-Seq and somatic mutation area, but also interested in the variants and DNA-Seq. Last week, I think last month, we actually had a webinar on another breakthrough technology. It's called cloud-based NAND, which I will also summarize a little bit today. And today we have a release on Array Suite 10.0. Early this year, which actually marks another landmark for this journey, QIAGEN acquires Omicsoft You guys probably have already knew about that. We plan to have another major feature release later this year, preferably, I think, as part of the group user meeting in September. So that will be the web access for the NAND technology. So this is quite a journey for the Omicsoft members and also for myself. I just wanted you to use this to demonstrate that for the last 10 years, Omicsoft and later on QIAGEN Bioinformatics will continue to listen to our customers and deliver features and data service for our customers. Array Suite, I think most of our customers have both Array Studio and Array Server, and we call it Array Suite. It is an enterprise omics data management platform. The user work on the client at this stage, mostly on desktop, sometimes on Mac desktop, but hopefully soon actually on web browser as well. And there's always a server behind this. This is called a Resurgence. I think most of our customers also have this high-performance computing that actually connected to the server, so that the server doesn't need to do all the processing job. We also extended this framework to cloud. I think some of our customers even have both. It is possible that you only have server or you only have cloud, but in our framework, it is possible to have both. And we actually recommend this type of mixed approach because sometimes it's convenient to have the local HPC. Sometimes it's useful to have the remote cloud. I think the criteria and the rule is very simple. It all depends on where where our customers actually put the data. If the data is on cloud, then the computing needs to happen on the cloud. If the data is actually on the local data center or your private data center, then it should actually be connected to the HPC. And our framework is actually open to both framework. And sometimes, if the user doesn't have a big data center or a big infrastructure, we also allow user to either use our hosted server or actually use use a powerful server to do the processing job. So this has been our framework, I think, since the release of Array Suite and also the release of our cloud extension. On the other hand, Omicsoft also works almost exclusively on the NAND technology for the last few years. So the NAND technology actually takes the public data and sometimes internal data as well, and put this into a NAND database so that all the internal data and public data can be integrated together. They are processed in the same way, and we focus on the summarized data such as mutation, expression levels, but still allow users to access to the raw data, for example, BAM files, or sometimes BAM summary files, what we call BAS files. And if there is a need, the user can always go back to the raw data and try to confirm actually the findings. So the NAND technology is really a breakthrough technology that allow users to normalize and to essentially to standardize the whole analysis process. But also all those hundreds of visualizations that we provide as part of the NAND is also useful for the users to generate insights from the data. So the data management component and the NAND technology component are really the two major focus of Omicsoft product. So now let's talk about what's next. I personally think Array Suite 10.0 is our biggest release ever. Not only because we introduced some of the most exciting technology such as cloud-based NAND, but also the improvement is essentially on almost every aspect. There are still, today I'm going to actually talk about like a selective 10 features that I think, was potentially interesting to more than just one customer. But I also will be talking about some of the features that are on development. They are not like 100% done, but they are actually ready for testing. And they will be finalized before the user meeting. But if the user wants actually to use it now, they can. So this includes Sentinel integration, IPA integration, HCMD integration. Those will be described after these 10 features. So this cloud-based LAN, from my point of view, is the biggest breakthrough actually after we introduce LAN. It's when we introduce the LAN, we think, oh, this is a vector database. This is a very novel way to handle genomic data, and it's probably the only way to handle genomic data for data scale such as TCGA. But when we started to process more and more data sets, we found that the previous way of doing LANs actually have their own limitations. The biggest problem that I personally found, and I feel, oh, this is really is going to kill us in long term, is actually the time that's needed to deliver the LAN update. Sometimes it takes 36 hours. Sometimes it could even be longer, depend on the internet speed, and also depend on the user's hardware. But we cannot just continue doing more and more data analysis, because that's the whole point of doing LAN. I mean, the LAN itself is a technology, but eventually it's data subscription, right? So at this stage, I think the framework needs to be significantly improved, not just to improve the delivery time, but also really to make sure that we can easily process, deliver, but also search the data that's potentially 10 to 20 times than what we have now, which I think will happen in 5 to 6 years. So we potentially, right now we have about 1.5 terabytes of data, but I think in 5 to 10 years, we potentially can have 15 or even 20 terabytes of data. So this is, cloud LAN actually solved all of these problems. So the performance, which I will actually demonstrate in the next slide, was significantly improved for both delivery and search. It actually provides automatic updates. So the user doesn't need to worry about the update at all. It happens actually on Saturday night if there is an update. And of course, the administrator can control that process. And we guarantee to have at least one update every quarter. But if there is a need for some specific LAN, we can actually have more frequent updates, such as monthly. If there is like a strong interest, we can even do weekly updates for some of the LANs. Scalability, that's why I mentioned, it could actually, what if we actually have 10 times more of the data than we already have now? Cloud LAN actually solve that problem. So let's take a look of the combination of 10.0 and cloud-based LAN and see how actually the performance looks like. So this is for searching, which is probably the most common operation for the user. When they get to the LAN, they search for either one gene or multiple genes. So previously in 9.0, for TCGA_P37, which is definitely our biggest LAN, connecting to the LAN could actually take 15 to 20 seconds, depend on each company's different situation, hardware and also internal bandwidth. This always happens. Like no matter how you actually search the gene, you will always take that long for any gene that you have. Now for 10.0, if you want to switch to another LAN, it only takes 4 to 5 seconds, and this of course only happens once. And if you search for a single gene, previously with all the overhead and other things, it could take actually 20 to 25 seconds in the worst case. On average, I would say 15 to 20 seconds. But now, you actually have two cases. First, if you actually try a gene that nobody in the company ever tried before, it will be 5 to 6 seconds. But if you search for a gene that's already been searched by someone else, which means that it's already been cached from cloud to local, it's actually 2 to 3 seconds. In practice, it could be actually 1 second. It could be so fast that you don't even actually realize it's already been the search is done. Previously, if we try 100 genes for TCG_B37, it will take more than a minute, 75 seconds, sometimes even 2 minutes, 3 minutes. Now, 15 seconds, which is 5 times faster. You actually get that gene, 100 genes searched. So to me, this is really important for advanced users who actually have to search genes, like almost on a daily basis, but also they need to search, search different genes and different lands and need to do a lot of searching. But in practice, this actually will improve the user experience. And as I will mention in the next slide, if this kind of search is actually happen on the from the API, from the programming side, this actually will make an even big impact. So so land has a lot of advantages, but there are actually still a lot of potentials that I think the cloud land can provide. So I'm just listing a few things here. When we apply this cloud land technology to genetics, TCGA Gemini, which is now finalized and generally available for users who have access to genetics land, it can actually publish in 5 minutes. And this is actually a VCF file with 7 million variants and more than 10,000 samples. The VCF.gz file, which is already compressed, is actually 267 GB. Essentially the biggest VCF that will ever generate. This is joint core based on GATK. Previously, downloading this file and start to publishing, we never tried it, but I think this is not something that can be done in 48 hours. Now it's 5 minutes. It also allows merging thousands of associating reports and allow us actually to deliver this associating report, allow the user to search through these thousands of associating reports. And also because we have this access control built in for Cloud Lens, if the user have access and have co-application with us on TCGA or other land, other projects, it can be actually, this full control can be configured in a few seconds, and the user will be able to access these restricted data sets. Another advantage of Cloud Land would be variation control. Some companies actually like different versions of the statistic analysis, so they prefer to use WOM instead of D6-2. Sometimes, for Mercury data sets, they don't want to have general linear model. They say, oh, I actually like Lima a little bit better. I want all the data sets to be processed by Lima, unless it cannot be processed by Lima for some complicated models. But each company actually has a different combination of requirements. For us, after we actually get all the data ready and we get all the metadata ready, we don't really care like which method the user prefers. But we need to provide this kind of variations for the user easily, and Cloudland actually perfectly solve that problem. So if the company, the administrator from the company says, oh, we want WOM, then we actually just change the configuration and provide WOM. If the user wants to say, oh, I want Lima, then we actually just change the configuration based on the company name and make that change. So previously, it will actually involve a lot of delivery for the user. Now everything was controlled by a single configuration file. So the last but not the least, the web access based on this cloud-based NAND, I personally think will actually make a major impact for the users, especially for advanced users, because it will allow users to access NAND subscription data, which now is more than, not actually including genetic NAND data. It's already 1 terabytes summarized data, so those are the expression matrix and mutations, copy numbers, those type of things. If we actually provide user RESTful API, and the user, is only interested in data that we provide, the user doesn't even need to connect to the server because cloud has it. We provide the user API key, RESTful API, the user can access this 1 terabytes of data easily. So so that's really the from Omicsoft point of view. This is a lot of you guys already have some ideas from Gary's webinar, but I want to reemphasize that this new technology plus this optimization that we did actually on 10.0 is going to actually change the user experience for that. And this is why I think this is the top one feature from the new release. So the next one I want to talk about is single-cell RNA-Seq support. We have been working on this for over a year now, and pretty much every company is starting, has already started this this new technology, or actually about to start this technology. So there are lots of things going on with single-cell RNA-Seq. So I think we have achieved a lot, but I also want you to be clear on what has been available and what has not been available. So we can't, with 10.0, which will be released at the end of this month. I mean, I will talk about the timeline a little bit later on. But the plan for this 10.0 release is that we will expose and we will continue to refine these modules on preprocessing, filtering, QC, code alignment, single-cell counting, compressed data storage and manipulation. So when you have actually a large sparse matrix, what is actually the offer from our product, but also allow users to import and export if you already have another pipeline that generates this single-cell count data. So what's not being supported yet actually are those state-of-the-art class algorithms. So we have seen a lot of new algorithms that are designed for this single-cell data. They are usually available in open source tools. We still have plans to actually integrate these algorithms through our engine, so that the user can easily get this clustering through the server-based integration. And additional downstream analysis, some of the new paper, new methods that are interesting but probably are not so mature. At this stage, they were provided more or less based on our integration or eScript integration, not based on native implementation. Because one thing that we found is that because the technology is so new, it's very hard to try to re-implement those algorithms that sometimes are not even mature. One thing that we think is important is the servers to predict the cell types based on cell signatures. So basically, this is more like a data service from QIAGEN. So we actually build a database of cell types and the signatures. But then when the user has the single cell count data, when they have like thousands of cells, we provide predictions for each one of the 1,000 cells for each sample. Not that they actually get the cell type and they estimate the mixture, probabilities or mixture proportions of the cell types for the single cell. They can do easy comparison. Compared to algorithm, this is more like a supervised approach instead of unsupervised approach in TSNE or CETA. So let me actually talk about the modules that we already supported. We think that pre-processing of single cell is important, alignment is important, counting is important, and this is essentially done. The pre-processing of different technologies, including 10x genomics or other job-seq technologies, was actually implemented in this general module that allow users to specify those barcode as a regular expression. This applies to 10x genomics, but also applies to other data sets. So our goal is to be as general as possible. So no matter what kind of data or how many actually FASTQ that you have for a single sample, you can still use our module actually to do the pre-processing, which actually generate this kind of tag files. The tag file will include, always include the cell barcode, but sometimes they also include UMI barcode, qualities, and others. And these kind of tags, they are important because we want these tags to be part of the BAM file. We actually need to generate these tag files at first because the tags can be part of the read names. It can be part of the sequence. So we need a standardized way to generate these tags. And then we have another module to filter out these barcodes based on whitelist. We actually have allow users to remap the barcode. We also automatically correct some of the barcodes based on sequence area and clustering algorithms. This so that the user actually will have all this kind of pre-processing that they expect to see from the original sequence files. So this could be complicated for users who are new to this technology. So we provide this kind of wiki page. It's called pre-processing of single cell RNA-Seq. Right now we provide 4 different cases, but I imagine that based on the user's data, we'll provide more and more actually examples so that the user actually know, based on the pattern of the data set, what kind of pre-processing is required. But then as long as the data are normalized and the tags are extracted, we actually provide a dedicated module based on our very popular RNA-Seq align module. But here, because we are generating barcoded BAM files, so which means that the cell barcode and UMI barcode and other qualities are part of the tag of each alignment. Well, actually, this module very similar to what you have seen for RNA-Seq alignment, but the goal of this particular module is to expect these associated tag files, which are actually the outcome of the pre-processing filtering module. But now we will automatically incorporate this tag in and generate barcoded BAM files, which actually will be the basis of all the downstream analysis. And then after we have this barcoded BAM files, which could be generated by Omicsoft, but also could be generated by 10x Genomics Cell Ranger, or could be actually generated by a user's own pipeline. We start this with barcoded BAM file, and the only requirement is that for those alignments that the user wants to count, there are some sort of like cell barcode, so that we can count by each cell. We can also count by UMI barcode. So in this case, we will generate UMI count, but it's not actually required. If the user doesn't have UMI barcode, or they don't want to have the UMI count, they can uncheck this. They can change the default tag length, which is CD and UV, to something else. They can also decide if they want to count multi-rays or not. In practice, we'll actually allow users to count multi-rays. If it's UMI, a lot of time it's actually stranded sequencing, so the counting module needs to handle that. So this is a very flexible and generally useful module. For 10x genomics data, we actually benchmark the counting with Cell Ranger 1.3. The results are essentially the same, based on essentially all the public data that we can download from 10x genomics data. So we know this counting module will give essentially same results as the Cell Ranger software. But I think the advantage here is that you can apply this to those data that's not from 10x genomics technologies. You can actually have the flexibility of choosing your gene model. You can also take advantage of the whole framework of the downstream data visualization, filtering, and exporting, which will be shown in the next slide. So the output of this counting module is actually what we call zero-inflated macrodata, because this is essentially what the result is. In practice, you see a huge matrix with lots of zeros. So it's not missing inflated. It's essentially zero inflated. The output data actually also include all the summary information that's needed, like what is the cell number for each sample, what is the for each gene, how many cells actually show the expression for this gene, and some other information. It's essentially a macro data, just like what you did from Affymetrix platforms, except here you actually have lots of columns, lots of samples. Oh, actually, I would say observations. You have lots of observations, lots of cells for each sample. So this is where it's important to have a fundamental support of this new data type, what we call single cell count matrix data. We actually provide this as a new omics data type, as you can see from this particular screenshot. So we are talking about 20,000 genes that actually have expression at least in one cell. Each column here, as you can see, it's a barcode for a cell, and each row here is actually a sample gene. We're talking about 272,000 cells. This could come from one sample before filtering, or it actually can come from hundreds of samples, where each sample has like over 1,000 cells. So when we have a data matrix like this, if we just use macro data, it's not going to be efficient, because the memory will actually kill most of the desktop. But here you can see that with the new sparse technology and compressed technology, can only use less than 200 megabytes of memory, and this is a pretty big action matrix that the user can work on. And based on this, newly support data type, the user can do subsetting, splitting, merging, exporting to different formats. They can sort, they can attach variables, they can attach observations, and most of the analysis, Mercury manual analysis, actually works for this new data type. Although I think in practice, when we have a matrix of this big, we think that the next step that needs to be done is to actually do this kind of classification and to simplify and to reduce the dimensions of the data, so that the average biologist can actually get the biological meaning easily from this huge data matrix. So, so that's about it for single cell RNA-Seq. As you can see, we're focusing on two things. First, we're focusing on helping user to generate this matrix from FASTQ files, which we think is a great step of a necessary step of data reduction. So no matter if you have 10x genomics data, no matter if you or actually something else, we have modules for you to pre-process, filter, align, but also count. So that's one thing. The second thing that we focused on was to provide this fundamental support of this large zero-inflated sparse matrix, and with actually very, very big dimension. Actually, we support this as a data type, a new data type in Array Studio. So that's the two things that we did for single cell RNA-Seq. The next one is actually ENCODE integration. So in last year's user meeting, we talked about that we are going to process all the ENCODE data, because ENCODE has just so many data, especially for basic biology research. So we actually did that, and we did that essentially for everything that we can access. Now we actually provided more than 60,000 tracks for human and mouse from ENCODE project, and we actually provide this not like other open source tools. We provide this in a fully controlled way, so the user can access every single. They can actually search and access these tracks from a user interface with the full access to the metadata. And as you can see here, from here, for this is human B38, you have more than 30,000 tracks to search from, but you can search by metadata. And this is really a lot of data processing efforts. So, for example, for human B38, they have over 2,500 actually BAM files. But when ENCODE project provides this BAM files, they don't actually have BAM.dai files, so they don't have index. So which means if you want to access to a specific ENCODE BAM file, you have to download it, and you have to run some software, which of course you can use a Array Studio to index that at first. But the problem in practice is that the user doesn't want to actually download a huge file. They don't even have space to do that. It will take a long time. They just want to stream directly. So Omicsoft took the effort of downloading these files, generating this BIM index, which is always faster than BI, and we actually hosted this index files from our website and improved our software to automatically locate to Omicsoft index files for the single BAM file. So the BAM files can be streamed. So for other data types, such as bigwig, which always include index files within this file, DFF files, which the sizes are manageable, and the peak files, I think those are not problems. So we actually provide automatic streaming on these files. But the point is that with ENCODE integration, the user can access to one or hundreds of these tracks of this 60,000 instantly. That's our goal. So basically, the user can use Omicsoft Genome Browser as actually visualization tool to any or all of the ENCODE data. The next feature I'd like to introduce, which I think is actually a very trivial in terms of effect, but I think this actually took a lot of effort from us, and we think that it's going to be generally useful. You can see that in 9.0, when you have multiple charts, especially for bug files and some other things, and when you have very long labels, it doesn't look very good. It seems very duplicated for those multi-level labels. But now you can see in 10.0, it automatically recognizes this is a duplication of labels, and we'll actually handle this. And the whole process actually can be controlled. So you can disable this feature and revert back to 9.0, which is probably not recommended. Another feature that I would talk about, because this is another feature. Say the user actually won't even, if I'm not talking about if the support doesn't talk about it, the user actually doesn't even realize that there is actually a technology behind this to make this improvement. So basically, we are seeing a lot of users start to actually stream BAM files and VCF files from public resource. This could be HTTP resource, or could actually be like a BAM file from Amazon S3 storage. So sometimes the connection between server and cloud, or the connection between the server and the web is slow, and there's nothing that we can actually do about it of that bandwidth. So we actually introduced this caching technology so that server will actually use like a page-like caching, although this is in file, to cache these cloud or web actually regions that have already been previously navigated. And we actually provide version control and automatic cleaning for old versions so that the space of caching was actually fully controlled. This actually works extremely well. The effect is that as long as the BAM file and the region was actually navigated once, you will actually get the performance as if this file is actually on the server. We provided new gene set analysis. It has been there for about 4 or 5 months, but we actually keep improving this process as part of the customer feedback. The new gene set analysis actually is a unique way to provide cross-land and cross-species enrichment analysis. So if you actually have not worked on the gene set analysis before, I encourage you actually to give it a try, because this is a way to take advantage of, I think, over 60,000, over 46,000, I'm sorry, 46,000 gene sets from our land subscription. So this includes OncoLand, single-cell land, disease land reference. The links land is actually, this is a database of treatment, thousands of treatments on the treatment data on the Mercury data set. So one of the nice things about this project is that it has a huge number of comparisons of different treatment and different time. So this particular line itself actually have over 27,000 gene sets that can be taken advantage of. This one, of course, requires disease land subscription. And also, this one actually provides, this is a cross-species analysis. They actually provide access to mouse gene sets as well. So this, at least at this stage, this is the only way to take advantage of cross-land and cross-species analysis. And this particular gene set analysis will improve over time as well. This is because it's one of our focus. The next feature is actually a cool technology. It's probably not so much used in practice, but I think we expect that it will be used more and more. Basically, Omicsoft provides a new technology to visualize and search hundreds of millions of rows, such as 1000G VCF file. But this is not just VCF file. It's also the annotation results from the VCF file, what we call OSCR. It can actually also be used to visualize a big table, which is located on the server as well. So this is an example of how this large table streaming works. This is 1000G VCF file. We're talking about 85 million of rows and more than 2,500 columns. With a table of this big, it's essentially used less than 50 megabytes of memory and works actually smooth on a desktop. So this is the power of large data streaming, but also the user has the full flexibility to search by list of regions or list of RSIDs to actually get the subset of the data they are interested in. So this one combined with annotation, which essentially is OSCR streaming, actually provide user a very fast way to filter millions of variants or hundreds of millions of variants into a few variants. Variable selection is another advanced analytic module that we think could actually have more and more usage in practice. So previously, we provide nice and easy ways for users to do single variant correlation. But in practice, when the user has both micro data and clinical data and other data, and they want to build an advanced analytic module, they wanted to do both variable selection and actually this kind of modeling, advanced modeling to predict future data sets. So this actually has more a lot of usage in clinical setting as well. We actually implemented the four most widely used methods is actually NATCH, which includes fossil, partially square, random forest, and symbolic machine to automate this process. This module actually works on Mercury data directly with any covariant that attached to the Mercury data. So it actually has a lot of common usage for advanced users. After the predicted model, we can actually do they can import a new data set, which could be Mercury data set, and predict the response easily with that model, or actually perform external cross-validation. The next feature I want to talk about is a very specialized transgene data analysis. We started actually this analysis about 2-3 years ago, but because this is so specialized, and we actually continue to work with interesting parties to customize this process. I wanted to say that for 10.0, we think that we have the final and ultimate solution for this particular analysis. The whole pipeline so advanced that basically all the user need is to provide the FASTQ file, and they will get a Microsoft Word report based on the analysis pipeline that we provided, with essentially every single analysis that they wanted from this transgene data analysis. And we provided a lot of novel technologies, which essentially are only useful for this analysis, such as secular alignment, cross-species fusion analysis, consensus analysis based on those unaligned rays. It's really a huge amount of work from the Omicsoft analytic team, and the analysis works so well that we think it's going to completely solve the problem. This one does actually require special license. So if you're interested in, please talk to Matt or QIAGEN sales team and talk about the capabilities of this particular analysis. So genome browser has been widely used by our users. We have actually started to do in 10.0. We listened to individual user requests, and we provided very advanced filtering and customization modules such as intron length filtering, soft clipping cutoff, navigations to allow users to move different exons, or even more tooltips on the exact location of different tracks. Those are just a few screenshots of that many things that we have provided. Plus, a lot of those very user-specific improvements, such as, can we actually have, when we share genome browser, can we allow users to keep the same ID so that the other users cannot see the changes next time they open the shared genome browser ID, or actually, sort the exon junctions in a way that the overlapping of the visible region, will be actually weighty, or actually when the user accidentally put a mouse down in a human genome browser, what's going to happen? So a lot of features that have been improved based on the user request. But I think no one's more exciting actually as this one. Coding sequence instant search. So if you have not tried this, please try this out, because this is my favorite feature in 10.0. When you have like a short protein sequence, just maybe a few amino acids, but you want to search through the genome browser to get like a bookmarks of this based on either visible region or actually the full transcriptome. Now the search actually was fully provided as part of 10.0. We'll also support fuzzy search. So it doesn't have to be exact amino acid. You can do actually fuzzy search with regular expression. And if you don't actually have have the genome because you are connected to a server and you don't want to download the whole genome, you can actually create a server-based genome browser and then connect to the server and ask the server to do the search. So a nice feature. requested by one of our advanced users, and we think that it's going to benefit every single user, at least genome browser users. So those are the 10 features of 10.0. I also want to talk about some of the incoming features. Again, they are essentially done, but we either we haven't actually have done the full documentation, or we think that it's still at beta stage and we need actually more user feedback. But you can see that all those interesting features are coming. Sentinel integration provides 10 to 20 times faster variant calling, benchmarked with GATK, but actually much faster than GATK. So now if you have DNA-seq analysis that you want to go through GATK pipeline, and BWA, this is actually the user interface that you should consider. This particular screenshot shows you how you're going actually to make this, 10 steps, 10 steps of DNA-seq analysis variants, and actually get the golden standard results. You can start with FastQ, BAM VCF, and GVCF. And if you start with FastQ, you get everything. If you start with VCF, you get calibrated VCF. If you start with GVCF, we can actually do the joint cloning. And this is for the GMM mutation. We actually have another pipeline that will do tumor and normal, but also tumor only somatic mutation cloning. This was supported by Sentinel, but because the integration was so tight that the user doesn't even need to know that it was actually what the exact logic behind it. And I also wanted to mention that this particular pipeline supports both all the modes that we support, like server only, cluster-based, or cloud-based. IPA integration, we are now actually part of the QIAGEN bioinformatics team, so we can do essentially extensive integration with IPA. As you can see, not only will allow users to upload multiple observations, we also allow users to do advanced analysis options, to adjust this analysis, and we expect to see more and more integration. In fact, I already seen preliminary implementations of land-based integration for IPA. And this will actually, in the future, every studio users will also be able to automatically launch API and go to a specific view. So that's the IPA integration. HGMD integration, HGMD is actually a product from QIAGEN as well. We actually provide this full integration as annotations as part of the LAMP subscription and analytics. As long as the user has HGMD subscription, they can always just tell us, and we'll enable this from the server side, and all they need to do is to do one line configuration for the integration. So that's about it for the 10 features of Array Suite 10.0, but also some of the exciting integration features that we are working on. Array Suite 10.0 will be available at the end of this month. If the user wants to access the new version now, they can use the dev updating module. They can use the latest like online client version to access version 10. And if they want to change to 10.0, I think the best strategy is to install the new version actually on the dev server, which I believe most of the customers already have. But I think on May 29th, everything will be ready. All the documentation will be there. All the tests will be done. So I'm very excited about this opportunity to release a new version with our customers. Sorry, I'm a little bit late. We still have 5 minutes, Vivian.
Yeah, hi. Thanks, Jack, for the excellent presentation. And we got quite some questions. Jack, could you access to the questions box, or do you want me to read those questions for you?
Okay.
Well,
So,
quite some questions about the single cell RNA-Seq. Maybe you could start from the beginning of the question list and pick up some important ones that you think you definitely want to address, and we will definitely get back to every question individually later if your question is not addressed.
okay, I got a more technical problem here. And I can see a lot of questions. Yeah, the problem. Okay, all right. So I can see that there is a request for more details on the single cell modules. I think that would be the answer would be yes. Okay, so I've seen a lot of questions on single cell. I think we probably need another webinar on exclusively on single cell, which I think we can provide after May 29th. I saw a question for alignment step of single cell, but they require more powerful EC instance in AWS. The question is no. This is really just regular RNA-Seq. Even though in practice a single cell RNA-Seq with barcode alignment could be a little bit bigger. I think our standard requirement is fine. Someone asked how are we going to get the single cell RNA-Seq modules. The answer is to actually provide to use the dev implementations or wait until May 29th to get everything that we plan for 10.0. Is the extra license for single cell LAN? No. I think every user who have access to this LAN will be able to access single cell LAN, but also single cell gene sets. There is a question on Sentient for non-human genome analysis. I think Sentient was optimized for human and mouse, but I think for other species, because we don't really have those kind of DB SNP type of known variant analysis, I don't think it's optimized, but I think it's still, GATK itself is still a golden standard. So whatever GATK provided, I think Sentient actually will have that advantage. So there is a question on transgene analysis regarding support NGS sequencing of antibody libraries to support. I think this is a very interesting question. The customer who actually answered this question probably should contact us directly, because there are so many novel technology we developed, such as word reporting for this transgene analysis. I think it's very possible that we can provide similar service for antibody libraries.
And here's a question of can we provide a recording? And yes, we will provide a recording and send to array attendees through emails shortly after the presentation.
Okay, and the last question I would like to touch base is a question. There is a question on the plans to expand the support to other omic data other than genomics and transcriptomics. I think in practice we actually work mostly on genomics and transcriptomics, but most of the modules also works on proteomics and other data types. So if the user, and actually we are the golden standard for metabolomics as well. So if the user can ask more details regarding what type of data, we will be happy to discuss these opportunities. So I think that's it. Yeah, but again, I think we will have reading replies for every single questions over the next few days. And anytime if a customer have specific questions, please feel free to email me and Matt directly. We will either reply in person, or we will make sure someone in the support group reply to the customers. Okay, again, thank you very much for attending this meeting, and we are very excited about the new release, and we hope to finish this release very soon, and get every all these new features to you as soon as possible. Thank you.
All right. Thanks very much to Jack and everybody for attending this webinar. And please stay tuned as we will possibly host a webinar about the single-cell RNA-Seq. And please also stay tuned for our upcoming user group meeting, which we will be ready for registration in a couple weeks. And again, thanks for Jack and everyone, and we'll hopefully see you next time. Bye, everyone.